Hi, now we want to discuss about the final part of engineering immunity, but this is a bonus as we're gonna talk about innate and adaptive immunity. This is a review. So at the start, I want to show you this a very interesting movie again for tracking of mobile cells, which is immune cells here in the dermis, which is underneath the skin after skin irritation. So what you can see over here, these are big round ones, are hair follicles. Outside is a tissue containing like blood vessel and connective tissues. And what I want you to focus is for this 25 minutes, little faster movement, how hard and how diligent our immune systems and cells are working. So let's take a look. You see these little cells, and these immune cells are working hard over the time. So they come back and forth, come in and out of a blood vessel and inside tissue. So looking for, I think this is a process kind of inflammation. So whenever there is any insult, in this case, skin irritation was done by putting a tape and then detach. So over this, there's some minor skin irritation, and our immune cells are actively responding to it. So what are the differences between innate and adaptive immunity? So first, let's start from innate. So innate immunity is to help destroy pathogens, including like bacteria or virus or something else outside of our body before they can infect our whole cell. So this is really the first line of defense. That means very early stage, and so our body immune response should be fast. And at the same time, this is a kind of a non-specific, meaning that we only recognize of very common molecular component, mainly expressed or shown by, let's say, bacteria. So for example, is LPS or lipopolysaccharides, and these are gram-negative bacteria outer membrane have this component. So our system, probably immunologically developed, our innate immune cells such as macrophage or neutrophils will see LPS, then they directly attack this external attack. So in that regards, so this innate immunity do not have a memory, which memory means once you have an invasion from outside, you remember that so that we can prepare for the future insult. And in the future, we gain faster and faster. But this innate immunity is irrelevant for the memory. So it's more related to when these invasion happens, we produce a general inflammation, which is representative by like 4 phenomena. One is heat, so the temperature goes up, you all remember, and vasodilation, which is a enlargement of the blood vessel, so that, and lymphatic vessel probably, so that the vessel will dilate, and also it involves the pain. So we know that there are something happening there. So it gives a signal to our brain and ourselves, and also because of probably vasodilation, and there's a swelling, which is an exudate of a plasma into the tissue, so the tissue will swell. So these 4 phenomena probably helps us to protect ourselves from this invasion. So the component of this innate immunity, there are, I already discussed about a complement system, which starts from C1 to C9. And whenever there's an external one, it's inside this blood plasma, they see these foreign cells, the complement goes in and then make a pore so that it actually lyses those bacteria. So, and another component of innate immunity is white blood cells containing macrophages, neutrophils, and NK cell or natural killer cells. So these cells are more like general innate immune cells. So what are the limitations of our innate immunity? Is they are very quick, they are non-specific, no memory, but they are not as always effective. And they may fail to clear the pathogen before infection is establishing. So we have a more elaborate system, which is slower, but adaptive to our external invasion. So here is a diagram showing primary adaptive immune response. So let's see the whole story, how things happen. First, this pathogen, such as virus, is coming into our body as an antigen. So that then, we have a macrophage recognize the pathogen and try to phagocyte us, try to engulf and eat up. Then these virus as an antigen will be broken down inside this phagocyte with a phagolysosome. And then those components, the parts of these virus will be represented out of this macrophage so that it shows that, hey, I got this one, I eat this, and these are the parts. And the nearby, so macrophage presents components of the pathogen to the nearby T lymphocyte, T cell. So the T cells nearby, probably this can happen in lymph node. And then T lymphocyte has a very specific receptor. And when the key and lock is matching, then T lymphocyte, those specific T lymphocyte will be activated. And they will activate a killer cell, they will activate a B cell too. So the killer cells, or in this case, we call CTL, or cytotoxic T cell. So this cytotoxic T cell will now destroy those infected cells. So that's one way the killer cell, cytotoxic T cell, will recognize those the same kind of a antigen. They will go and they will kill. On the other hand, this also will activate a B lymphocyte. That B lymphocyte is also very specific to those specific antigens. So they will have a specific antibody against that antigen. Then that specific B lymphocyte will start to proliferate. So that into plasma cells, so like clonal expansion of making many of their clones to produce the same antibodies against these antigens, so making many antibodies so that this antibody can attach and label these antigens, which you already remember that opsonizes this, such that macrophage prefers to phagocytose these ones, and also neutralize and many other functions by these specific antibodies. On the other hand, the B lymphocytes, some portion of these B lymphocytes were differentiated into a memory cell. So that memory cell is not for now, but for the future. So they later induce secondary immune response upon renewed contact with the same pathogen. They were like soon as a kind of reserve forces specific to this antigen, we have now memory cell. So that's probably when we do vaccinate, we will have this multiple of these memory cells available so that we can protect ourselves from future attack of these specific viruses. So now let's discuss and summarize this adaptive immunity, which is an acquired immune system. As an example, this contains APC, or antigen presenting cells, and B cell, T cell. So these cells are the main component of our adaptive immunity. And as I said, they are not first line of defense, but the second line and the for future. So they are usually slow and they have memory effect because that's for protecting ourselves from future reinfection. And adaptive immunity can be composed of two parts. One is humoral immunity, and the second is a cellular immunity. Humoral immunity, the humor is more like what's happening in blood plasma, while cellular is a cell-mediated immunity by the specific adaptive immune cells. So let's first humoral response means the B cells secrete specific antibody. I mean, specific means specific against those specific antigens recognized by our antigen presenting cells. So that antigen, it means binds to antibody or receptors on T cells, and then that specificity will be activate B cell by T helper cells. And cellular response, the second is cellular response, and that's when pathogens that actually can evade our innate immunity, and they may succeed, in fact, host cells, then those infected cells can be phagocytosed by professional antigen presenting cells, such as dendritic cells and macrophages, and that's related to our cellular response. So let me go a little further to about immunological memory formation by B cells. So the B cells finding out the bacteria, which is an antigen that matches to its receptor. As I said in clonal, the antibody diversity, which means also the same as B cell receptor diversity, because that receptor is the antibody, specific antibody. When B cells residing in lymph node, they got this bacteria or lymph node or inside tissue, that bacteria has an antigen specifically fits, that B cell now will be activated, and the helper T cell around will activate this, making the B cell to proliferate into, differentiate into plasma cells and memory cells. So those plasma cells will also produce antibodies that attach to the current type of invader, and they will also proliferate to generate many antibodies. Because of these antibodies, those phagocytes prefer these marked pathogens with antibodies and eat them. And at the same time, simultaneously, memory cells will remember this. And when the same intruder will come in the future, this memory cell will help the immune system to activate and respond fastly. So let's, this is final for this review and phases of response to an initial infection. So let's focus on the timeline, which is important for us to know. So when there's an infection happens, we need an immediate first line of defense as an innate immunity, that is a recognition by preformed, non-specific, and broadly specific effectors, such as we see LPS, then we recognize it and kill this, remove these infectious agents. So an early induced any innate response involves, let's say, from 4 hours to several days. Then now it's recognized recognition of this microbial associated molecular parent, then it causes inflammation. You remember that you've got, let's say, some hit or a small cut, and you have inflammation. So, and it lasts for several days. So this inflammation recruitment and activation of effector cells that's coming in, and then removal of infectious agents. And now after several days, let's say more than 96 hours, let's say about a week or after, now adaptive immune response will take part. So the infection and that transport of this antigen to go to the lymphoid organs such as lymph node, and then there's a lot of naive B cell and T cell, some of them will recognize this specific antigen. So recognition and those specific B cell and T cell will have proliferate. We call it clonal expansion, because they will generate exactly the same antigen, the same type of T cell receptors, and they will differentiate into an effector activated cells and B cell and T cell, and they will remove infectious agents like cleanly over time. So as a summary, so this engineering immunity section, we learned about the role of vaccines in the prevention of disease. And especially we study about the antibodies in the immune system and ways how to use antibody to prevent diseases and even treatment of disease, so that we learned the engineering of antibodies, like there are human ones into animal-grown ones and humanized antibodies, etc. And so that we understand the antibody structure and different kinds of antibody classes. And we talk about the differences between monoclonal antibodies, which could be used for therapeutic purpose and research purpose, and also polyclonal antibodies, and how to manufacture, create monoclonal antibody, which led to Nobel Prize, and requirement for effective vaccine development. And finally, we have a immunology 101, or the basics of immunology, such as innate and adaptive immunity. Thank you for your attention.